Welcome to all our guests, and thank you for participating in our webinar on computational medicine. This webinar is has been organized by our Italian friends. Our president, Elisa Bianchi, will now say a few words, and then our experts on computational medicine will have their lectures. After that, we will open to the question and answer. We'll have a time for question and answer. You can raise your digital hand. You have a button on the bottom of your screen, and I will open your microphone so you can ask the question. If you can ask it in English, perfect. Otherwise, you can ask it in Italian, and we can translate it for our experts. So I think, Elisa, please, the floor is yours.
Good afternoon, everyone. As president, I offer the greetings of the Italian Association Amici dell'Università di Gerusalemme, Italian friends, and thank all those who participate and are connected. In particular, Professor Ben-Yehuda, Arzy, Carmi, Elli, Basi, and Professor Ittai Ben-Porath will manage the event. The occasion is important because we talk about the medicine of the future, and all of us are eager to learn. Good work then, and I leave the floor to Professor Ben-Porath. Thank you.
Yes, good evening, everybody. I hope it's a nice Sunday, wherever you are, in Milano or in Italy or anywhere else. We're speaking from cold Jerusalem today, and to discuss a little bit about computational medicine. What is it? What does it mean? We have with us, so my name is Ittai Ben-Porath. I'm a professor at the Hebrew University Faculty of Medicine. I study cancer, and I'm a vice dean helping in the development of the faculty. And we have with us our dean, Professor Dina Ben-Yehuda, which is also a chief of hematology and an expert in blood cancers. The two other speakers that are experts in medicine and computational and theoretical approaches that will tell us about their work are Dr. Shahar Arzy, who is a neurologist, treats patients, Alzheimer patients, but also is doing pioneering work in employing computer approaches and machine learning approaches for treatment. And Professor Shai Carmi, who of our School of Public Health, which is an expert in genetic and genomic approaches in medicine and public health. And, we also have with us Marco Cannella, who is a PhD student in our faculty, and he's from Italy, and he's helped us prepare for this evening by adding some Italian to our slides. He can also help with questions and translations. And so if you have questions and you post them in the chat, either Marco Cannella or Marco Elli or any other one of our participants can help in answering them. And if we need, and it's hard to follow, we can stop and answer the questions. So I think at this point, Professor Dina, so we're hoping to do this in the next hour. So each of the talks will be 10 minutes or 10 to 15 minutes, and leave some time for question and discussion at the end. So Professor Ben-Yehuda, please, the floor is yours.
Thank you, Ittai. Buonasera. I would like to present to you our mission and our goal in the past few years and in the future. We would like to lead the global revolution in computational medicine in the era of personalized digital medicine. And you can see here the translation that were done by our student. I'll wait a second. So our faculty is composed of 5 schools. We have the medical school together with the military medicine track, which is called Tzameret. We have nursing school with the military track, which is called Pisga. We have the School of Pharmacy, School of Occupational Therapy, and the Brown School of Public Health. We also have a biomedical science program for students that are studying preclinical studies. In our faculty, we are investigating 5 disease-oriented translational research programs, including cancer research, infectious and including tropical diseases, metabolic diseases, neurodegenerative diseases, and stem cells and regenerative medicine. And again, you can see the translations here in the bottom. So medicine is undergoing revolution in the past few years, from a medicine that was like trial and error medicine, where one treatment fits all. We are now trying to do precision medicine to each and every patient. And the easiest way to understand precision medicine is to understand that its emerging approach for disease prevention and treatment that takes into account individual variations, not only in his disease, but also in his environment, lifestyle, and genes. And to be clearer, what does it mean? So it all starts when a patient comes in. We need to take a careful clinical data, tissues, and understand the basic clinical picture. But also we look at the genetic data and data that is called epigenetic, when we are looking at genes that are being expressed in a given patient. We look at biomarkers, but we also take into account environmental factors, diet, and the microbiome. The microbiome is the content of bacteria in our body. We know now that this bacteria can dictate protein expression in our body. In this way, we hope to have personalized diagnosis, personalized treatment, and moreover, personalized prevention. So what do we need for it? And I'm happy to say that we have it all in our faculty. First of all, we need translational research to understand the mechanisms of the disease. You can see it here in Italian. I hope that it's clear. But if we want to understand what's the difference between our patient and, let's say, thousands of patients with the same disease, we need big data. We need many data, many parameters on many patients to understand what is the uniqueness in the patient that sits now in my office. For that, we need information technology. And as you well know, in the Hebrew University, we have it big time. We need bioinformatics, and we need computational medicine, and I'll come back to it in a second. But in the end of the day, we also need somebody that will develop drug and technology depending on this data that we'll have about our patients. So what are we doing in our faculty as for preparing the next generation of physicians in the world? So first of all, I believe that physicians, research physicians, bring to the lab things that the preclinical or the basic scientists do not have. First of all, we understand the disease. We touched it. We know the patients, what he's suffering from, how the disease looks under the microscope. Sometimes we participate in the operation where we can look at the disease in our own eyes. So we understand the disease better. The other thing that we bring to the lab is an unmet need of a given patient. On each and every research that I did in my lab, I can tell you the name of the patient that forced me to come in and to do the research in order to solve an unmet need of a given patient. So we have a very strong track for MD, PhD students, students that we hope that will be research physicians after their graduation. And we have 89 students like that in our faculty. The second thing that we give a lot of emphasis is to educate more students that will be computational physicians. What do I mean by that? Physicians are very preservatives. It's very hard for us to change protocols, to change the way that we are thinking. And we are very concerned that once the technology of machine learning and AI will be here, the physicians will be too scared to use it and to apply it to the day-to-day clinic. So we created a track, which is called Sagol track, because Sami Sagol donated a lot of money for this program, where very, very bright students are studying together both computational science and medicine. So we'll have physicians that will talk two languages, medicine and computational science. And I hope that they will be like the ambassadors that will bring the new ideas, the new technology of using AI to be better physicians. So this is the group. We have 60 students now. You can see how many girls we have here. And I'm very proud of each and every one of them, because they are really studying very hard. You know that medicine is a very complicated and very hard studies, and they are studying both computation science and medicine. We also created another course for a master degree in MBA Biomed, where we are developing students to work with innovation and entrepreneurship. And here we have also 35 students. We just started this program. And in NPH, we have 59 students that will be both physicians, and as a physician, they will work in epidemiology and in public health. And the new buzz now is bioconvergence. What is bioconvergence? It's the place where very developed biotechnology meets the very developed software and engineering, and creates a new subject of bioconvergence, which is like all the robotica that we use in medicine, the AI-driven remote monitoring, organoids where we can take one cell from a patient and develop a whole organ, and organ printing. So our aim is to create a unique working environment where after we'll collect all the data and we'll do data processing, the research physician, the basic researcher, and this little one is our computational physician who has a white coat and sits in front of the computer. And in the end of the day, we hope like this to get personalized treatment for Noah. Why Noah? Because this is the most common name in Hebrew. And Ittai will talk with you more in the end of our meeting about the building that we are building now in order to have this unique working environment. So many times I'm asked, will AI replace physicians? So I'm sure that it won't happen. Moreover, I'm sure that in the digital age, patients will need more compassion than even now. And whenever I'm putting the stethoscope on the heart of my patient and I'm holding his hand to palpate his pulse, I feel that it's not only that I'm listening to the heartbeat and murmurs, I listen to his heart. And this connection, this touch has a lot to do with good medicine. And I believe that no computer will take this place. I know that when I'm palpating the abdomen of a patient of mine and I'm looking at his face to see when it hurts, I'm sure that this is something that only human will be able to do. So AI will make us better physicians, but compassion will still be needed always. And we are teaching now compassion in our medical school via end of life and palliative care medicine, integrative medicine, interprofessional medicine, but also patient safety and quality of care. We give platform to our students where they can write about the reflection when they meet with patients and so forth. We have simulation center and also gender medicine. And I thank you for your attention, grazie. And I'll be happy to answer questions later on. Thank you.
Thank you, Dina. And we will have all of the questions at the end. So I think we will continue now to Shahar Arzy, who will tell us if we need both artificial intelligence and the actual physician working together. Shahar, please.
Yeah. Okay. Yeah. Good. You see my screen with the presentation in Italian?
Yes, we see it.
Yeah, and can you hear me? Good. So let's start. So buonasera, signore e signori. Mi scuso per non parlare in italiano, però uno giorno. I would like to share with you our work in the lab about how computational medicine can help in preventing Alzheimer's disease. I'm, first of all, a cognitive neurologist, and I see patients, half of them are suffering from dementia and Alzheimer's. And working with this population and being unable to give them a real treatment, so I decided to dedicate my efforts to find a breakthrough to Alzheimer's disease. And I moved to the university, to the Hebrew University, and with the help of our brilliant students who are really the best genius available now in the world. They are coming to the Hebrew University, and they are also the most idealist ones because they want to do something significant, something meaningful. And we are trying to have a new approach to Alzheimer's disease through computational medicine, which I will present in the coming 10 minutes. So first of all, just 2 minutes about what is computational medicine. So a good definition that I personally like to pursue is that computational medicine aims to advance healthcare by developing computational models of disease and personalizing these models using data from patients so we can have a specific treatment for a specific patient, for the individual patient, because we are all different. And even if we are suffering from a disease that has a common name, the way in which the disease is expressed in the patient is different. Finally, so we apply these models to improve the diagnosis and treatment of the disease. So if we take an example, so here you see a model of a neural network model at this, that's in this example. And the model gets an input. The input can be clinical data, demographics, lifestyle, genomics, whatever you like. Everything can be fed or inserted into the model. Then the model does something, and we get outside from the model, we get diagnosis or prognosis, like how the disease is going to behave, or a pharmacological treatment or non-pharmacological treatment. Now you can ask, what is doing what the model does? So the revolution of computational medicine or the revolution of machine learning actually starts with the assumption that it doesn't really matter. Like if you take this bambino here, and the bambino sits with his mother, and the mama teaches him, for example, to do this game. So she gives him examples, and then when he does well, she says, bravo, bravissimo, kol hakavod. This is a feedback. Now we don't know exactly what happens in the brain of this baby, but nevertheless, we give the examples, we give the feedback, or even we don't have to give the feedback from time to time. Sometimes the baby himself or herself sees that she made it, and then she is happy with it and gives herself the feedback. This is unsupervised learning. So the model does the same, and we don't have to understand everything. We put the mathematical model, and if it functions well, we are happy. So this is a 1 minute about machine learning, and Professor Shai Carmi may agree with me or not later on. Now, what about Alzheimer's disease? Some numbers. First of all, presently there are 40 million patients with already Alzheimer's dementia worldwide. And in 2030, which is next year or close, it should be 55. And in 2050, it almost triples itself to 100 million. Now, most of the effort to develop treatments against Alzheimer's disease are related to biological treatments. And this is, you can see here, Biogen is one of the, is a big company and was the leader of Alzheimer's disease, modifying disease treatment developer. And you see here, like less than a year ago, when in June, when they announced that they have a drug that got an FDA approval or that was successful in a trial, and you see that a company worth, instead of 270 billion, they almost doubled itself to 450. But since then, when the drug was really used and people saw that it doesn't work, and the company went down. So this is just an example that we are so far failing in treating Alzheimer's disease. And that's why in our lab, we thought that we have to think about it in a new, fresh manner, and the way that that the world goes in the last 40 years is a wrong way. So let's get an example. And the example is not very heroic, but is very impressive. And this is from heart disease in the last 40 years. And what you see here, what was the death rate per 100,000 people at the 70s, and it was more than 1,000 for 100,000, so more than 1%. And you see that it dropped and still keep dropping to the fifth of it, yeah, by 2017. Now there is no drug to heart disease, but to heart disease we know that we have to treat the vascular risk factors, and if people are training well, eating well, and getting treatment for heart for hypertension and for diabetes, and this will heal the heart disease, and there is no heart disease pill. So our idea was to do the same for Alzheimer's, and then we faced 3 challenges. The first challenge is characterizing AD. What is Alzheimer's disease? Causal Alzheimer. The second challenge is to do early identification of Alzheimer's disease, and not only in the hospital, but also for the public, how we can reach the public and to do early identification of Alzheimer's disease. And this will be, in a second step, critical for applying preventive interventions before pathology advances, because also this heart disease, one cannot come with a heart attack and then say, okay, now I will stop smoking and I will stop eating sugar and calzone. It has to be started early on. And the third challenge is precision medicine to prevent Alzheimer's disease. So this is a long story. I have a course on it of like 24 different classes, but we have only 7 minutes. So let's just mention some important things. The first thing is what is Alzheimer's disease? So people usually, if you go and ask people what is Alzheimer's disease, people will speak about memory. So Alzheimer's is a memory problem. However, when you see people with Alzheimer's, most of them, they have memory problems in a later stage of the disease. So the advantage of the machine learning is that the machine learning is free of assumptions. It comes fresh to the data. You need an algorithm, and the algorithm is mathematics, and you can check the mathematics, and you need data. So this is an example. When you take a lot of data, it's a PET imaging data of tau tracer. Tau is a protein that's related to Alzheimer's disease in thousands of people. And then you take a simple algorithm and ask the algorithm to classify the data to different groups. And we do find one group, almost 20% of the people, that the pathology is in memory regions. However, 70% of the people are in another region. And there are also 2 small subtypes or phenotypes of Alzheimer's that we know from the clinic that are related to language and to high visual characters. So the question is, what is this behavioral phenotype or subtype of Alzheimer's disease? And what we claim is the behavioral phenotype relates to disorientation. Orientation is the relation in between the humans and the world, the space around us, the times, the events that we live our life within, and people that we live with. And you know that this is actually what happens in Alzheimer's disease. People lose their way, people lose their orientation in time, they don't remember, it seems that they don't remember, but actually what they don't do is they don't relate the different events to different times. And also they don't know how to manage those social network, and they don't identify people, and then it becomes very sad at the end when they don't identify even their grandchildren. For me, this is like one of the saddest moments with patients. So first, we define that behavioral phenotype of Alzheimer's is orientation, the relation between the patient or the person and the world. Now, how can I convince you this? This is the map where this is a brain in a pet where amyloid, another protein that related to Alzheimer, is depositing in the brain. And this is the brain system that we found that manages orientation. And what you see here, that the same system that manages orientation is a system disturbed in Alzheimer's disease. So we wrote an app and a computer application. It even has an Italian name, Clara. And the idea of Clara is to do several things. First of all, to do some games in order to identify orientation, that people play with places, play with events, and then we can know how their orientation is. We ask them to give names of people in their life, for example, and then we ask which person or which place is closer to them. And also we collect different responses to different questions that are important for prevention of Alzheimer's disease. And you can see here, this is a performance of this little app. You see that it has 95% performance, with much better than other tests. This is a little bit of complicated, but these are the proteins that I told you, the tau and the amyloid proteins. And you see that, and this is a Clara scorer, and you see that Clara identifies all people with high tau that you see here, the high tau, and all people with high amyloid you see here in red. Only one dropped out, and only 4 people with without high amyloid or high tau were identified. So this is a very, very good results for a screening test. So so we defined what is Alzheimer's disease. This was the first challenge. The second challenge was to identify it in a mass screen. So you see here with this little app, we can reach very many people just with doing an application that takes some minutes, and we can identify if they are suffering from very, very early stages of Alzheimer's disease. I didn't mention that this is not for people with Alzheimer's. It's people that are normal. The neuropsychological tests are all normal, but when you do this app, you find very, very early stages. And then the last thing is how to prevent. So here we built a model, and the model takes into account several things. In the first level, the model takes the suspicion index, or what's the chances that the patient suffers from Alzheimer's disease. That's what I showed you before, that's the Clara. And then the risk factor score. There are 93 in the literature. There are 9, and we are identifying more, but there are 93 things that are related to developing Alzheimer's disease. Now, if a patient comes to me and I give him a list of 93 things to do, so he will just run away. So I have to find the 4 or 5 things that are more relevant to the patient. So one is the relevance, and another is the risk factor score. What is more important? For example, diabetes is much more important than hypertension. Like for heart attack, it may not be the case, but for Alzheimer's, diabetes is treating diabetes is much more important than treating hypertension. So this means that if I think that a patient suffers, there is a high probability that he suffers from Alzheimer's disease, I would like to treat very strongly even a pre-diabetes, an early stage of diabetes. So this is what the model does in level 1. In level 2, the model also.
Sorry to interrupt. I just want to ask you to take a look at the time, because we only have 1 or 2 minutes.
This is the last slide. So in level 2, the model takes what we just did and takes under account another more advanced features, like, for example, the disease progression or the dependency between the factors. And finally, in level 3, it also may take under account the genetic profile. And this is a project that we're supposed to launch soon with Professor Shai Carmi that will speak to you in 2 minutes. So the last thing is and this I'm most proud of this slide. This is people of our team that are presenting Clara in the world. Clara is already available in Portugal and it runs in Brazil, it runs in Boston, it runs in China, and you see it in very many places. And so you see Amnon here, and you see Tahel here, and Lidor and Greg, and really brilliant people that are working on this project. And yeah, and I thank you for the opportunity to show our work to you and to the people who are doing the work and enabling it, especially Professor Dina Ben-Yehuda. And thank you all so much. Grazie mille.
Thank you, Shahar. And we will continue now to Shai Carmi, who will tell us about how theoretical and computational approaches can be applied to for genomic uses in the use of genetics. Shai.
Hello, good evening, everyone. Can you hear me and see the slides? Yeah, okay, good. Yes, as Ittai said, I'm a professor at the School of Public Health at the Faculty of Medicine, and I work in genetics, specifically in computational genetics, using various statistical and computational tools in genetic research. And I will show you one interesting example of a new development in the field of genetics or genomics that led to a very interesting new technology, and I will explore the benefits and the possible problems with this technology. So we're talking about screening IVF embryos from humans for genetic risk of adult diseases. So let me start with the very basic introduction to DNA very briefly. The human DNA is also what we call the genome. It's a long sequence of a long chain of letters that has 3 billion letters. These letters can be A, C, G, and T, as you can see here. It's arranged in chromosomes, and these chromosomes, they appear, we have 2 copies of each chromosome in each of the cells of our body, and this sequence of letters, these are the instructions for the cell, and together with all the environmental exposures that we accumulate during our lifetime, the DNA and the environment, they determine our traits, how we look like, how we behave, and also what diseases we will have. So now I would like to focus on my subject, which is genetic testing of human IVF embryos. So when do we do it? Like, what are the circumstances when we have genetic testing of embryos? So this usually happens when parents carry mutations or changes in the sequence of the DNA for a severe childhood disease, sometimes very devastating or lethal diseases. I will give examples in the next slide. Sometimes it happens because of recurrent pregnancy loss or infertility. So the way it happens is using in vitro fertilization, or IVF, where oocytes or eggs are retrieved from the intended mother, and they are fertilized by the sperm of the intended father. And then the embryo grows in the lab, as you can see here, typically for 5 days, until it becomes a so-called blastocyst, as you can see here. And then a biopsy is taken, as you can see in this image. Just a few cells are extracted, and then in the lab, the DNA can be extracted. And then once we have the DNA, what do we do with the DNA, with the sequence of the DNA? We can read the sequence. So using all technologies, the clinicians used to look at just a single letter or just a single chromosome to see maybe a chromosome is absent, or we have an extra chromosome. But now with new technologies with kind of, the genomic revolution of the past 10 years or so, we're now able to sequence to read the entire genome, the entire DNA sequence of the embryo, and this is feasible and affordable. So this has huge implications regarding what can we test in the embryo. So this is really an emerging field. So as I said before, traditionally, the reason why parents would have to look at the DNA sequence of their embryos is to avoid very severe childhood diseases, like some of the ones I show here in this list. Most known is cystic fibrosis. These diseases can be lethal, and they're all caused by usually just one change in one letter in just one gene. So this is relatively easy to identify that change in the embryo. This is something that has been done for several decades now. But now the new technologies allow us to look at other diseases as well, which we call in different names. We call them adult diseases, or chronic, or complex, or polygenic diseases. Polygenic because they are affected by hundreds of genes or thousands of genes, not just by a single gene or a single letter in the DNA. And I provide here some examples of these diseases. So there was also, due to our ability to read the DNA sequence of so many individuals with such a low cost, in the recent years, there was huge progress in our understanding of the genetics of these diseases. And by now, we know of, for each of those diseases listed here and many others, we know of hundreds of genes that are associated with the disease. And beyond learning about the biology or etiology of the disease, now we can also use these results to predict the risk of individuals. So for each individual, we can compute something called risk score, or genetic risk score, which can be computed based on the DNA sequence of the individual, and it will predict the risk to become affected in the disease. So this is an example for coronary artery disease, or heart attack. So what we see here on the y-axis is the risk, is the probability to have a heart attack, and versus the percentile of this risk score, meaning individuals who are here, they have risk at the bottom 1%. So they have the lowest risk. And individuals here, the 50th percentile, they have the median risk. And individuals at the top 100 percentile, the top 1% of the risk, they're very high risk, and there, as you can see, their risk to have heart attack is something like 5 times higher than the average in the population. So these risk scores now have the ability to predict the risk of individuals to become affected in adult diseases, and these are similar results for other diseases, and there are many other papers showing similar results for numerous other diseases. So what this implies, our ability to predict the risk, the genetic risk of a disease, implies, and together with the ability to read the DNA of embryos, implies that we can compute those risk scores for embryos that are not even yet born, not even yet transferred to the womb of the intended mother. And now that if we are going to have those risk scores computed for each embryo, we can also prioritize the embryos based on the risk scores. And theoretically, we can choose any disease or trait that we desire to look at and prioritize the embryo, select the embryo for transfer into the mother based on the expected risk for any disease of interest. And in fact, this is it's not permitted in many of the world countries, but it is legal in the United States, and there are also already a few companies that already offer that to consumers. In particular, Genomic Prediction is already up and running and has been doing that for a couple of years now. So how does it look like? This is an example from Genomic Prediction. Here they show kind of report cards for 2 embryos, 1 and 2 here. So this is male, this is female, which we know by looking at the X and Y chromosomes. They are euploid, meaning they don't have any missing or extra chromosomes. And now, so these are kind of standard measurements. But what we have, the new thing is now the risk for a number of adult diseases. So each embryo is shown the risk for each disease, as well as a combined health score for all the diseases together. Many people will look at it and we think, okay, this is really like science fiction, but a lot of people will think, oh, this is dystopia. And there are several ethical and social problems associated with this technology, which we reviewed in a recent article. So I'll go over them briefly. One problem is eugenics. That was a movement that was very active in the first half of the 20th century with the intention of improving the genetic composition of the population, encouraging couples that have good genetics, so to say, to reproduce. But very quickly, the focus became on discouraging couples with bad genetics to not reproduce, and later it led to concentration camps by the Nazis, and in several other Western countries also just forced sterilization of individuals with various conditions. So that was really bad, and since then any intervention in reproductive decisions is considered very problematic because of the chances of that it will lead to a slippery slope towards coercion by the states, and also just in general intervening with making those reproductive decisions about which lives are worth living is something that we may not want to do. There are also other problems like stigmatization of individuals who will be affected in the future by diseases that are people may see as preventable at some point. Also health disparity because IVF is very expensive, and there are several other problems. The work that I will describe in just a couple of slides is we understand pretty well the problems that there are with this technology, but we don't understand at all what are the potential benefits, because in theory this could also lead to reduction of the risk, to making the population healthier, making individuals healthier, and we wanted to quantify that. So I'll show you the results of this one paper. So we considered a mathematical model, and here comes the computational part. This is a statistical model that we solved using mathematics and using computer programs. So we considered selection for a single disease, and then we ranked the embryos. Each one of them is a schematic for an embryo. We rank them by their risk for the disease. And the way we select the embryo, we assume that we discard all the embryos that have all the embryos except the one embryo that has the lowest risk to develop the disease. And then our goal was to use a statistical and epidemiological model to quantify the risk reduction compared to selecting an embryo at random. In other words, not using the technology or not even using IVF at all. And then we want to see what is going to be the benefit of using IVF and selecting the embryo based on the genetic risk score. So the results of the modeling, I'll just show you one graph, one key graph. The results of the modeling show that are shown in this figure. Here, the y-axis is the risk reduction. So by what is the percentage by which we reduce the risk of the disease, and the x-axis is the number of embryos that the parents need to have in their IVF, in the process of the IVF that they performed. And now I'm showing you three graphs here. These two graphs correspond to realistic genetic risk scores based on current technology, based on current research. It's kind of like the lower bound and upper bound of their accuracy. And I'm also showing what we may have in the distant future, let's say in 10 or 20 years from now, when we have much more accurate risk prediction to have a certain disease. So this is not looking at a specific disease, just assuming that a disease that affects 1% of the population. And what we can see here that is very interesting, that even at 5 embryos, which is not a lot, definitely feasible, the parents can achieve risk reduction of 50%, around 50%, which is quite a lot and very difficult to achieve by any other means. So what we show is that this technology, despite the ethical and social problems associated with it, it does have the potential to lead to substantial reduction in the burden of diseases in the children in the next generation. But we do also have to say in parallel that these results are the best, represent the best case scenario. They are highly optimistic. First of all, they assume that the embryo that was selected based on the genetic score will be born. This may not necessarily happen. We know that it happens only about half the time. Sometimes it doesn't implant. Sometimes there's no pregnancy or there's miscarriage and so on. We also know that this is not going to work for women of advanced maternal age. Starting at age 38 or so, about half and then more than half of the embryos have chromosomal errors, so-called aneuploidy. And these women will very, very rarely have more than one live birth out of each IVF cycle. So selecting which embryo to use is not really a question. They will have to use every embryo they have. And even young couples, typically they will not have 5 embryos. This is optimistic, achievable, but optimistic, but typically they will have something like 2 or 3 embryos that will be born throughout IVF. Another problem, which is also very important, is that these risk scores have much lower accuracy in populations that are not of European origin. Most of the genetic studies were performed in the United Kingdom and the United States, and therefore these predictors are most accurate for individuals with European, mostly Western European ancestry, much less accurate in individuals of, for example, South Asian or East Asian or African ancestry. Even in Italian people, accuracy is slightly lower. In some populations, we don't even know what the accuracy is going to be. And this is again going to lead to disparity in access to healthcare. Other problem.
I have to ask you because we're really running out of time. So maybe
okay.
1 or 2 minutes and then
okay. Yeah, I'll finish in 1 minute, I hope. Another problem is that we don't know if the scores are going to be predictive. If we're trying to predict heart attack, we don't know if it will work in 50 years from now. And we're also afraid about that selection for one disease will increase the risk of the other. And then we have problems with communication of the results to the patient, the entire patient experience when they use this technology. Reducing risk by 50% may sound impressive, but when the disease is rare, for example, schizophrenia only affects 1% of the population. So if we reduce the risk by 50%, the absolute risk reduction is very small, only 0.5 percentage points, not very impressive when we look at it this way. Then these reports can be confusing and lead to stress in the patients. We saw a number of cases reported in the literature. The patients chose not to use any embryo because they were so confused. And doctors may not like it, and this can be very, very complicated. Just imagine that now a lot of people are going to wish to do IVF just because to use this technology is going to be extremely complicated. There are also many other unexplored issues with the way this is going to affect clinicians, patients, and their future children. So to summarize, this new technology that was enabled by progress in genomic technologies and also in statistical methodologies may be able to achieve large risk reductions in the next generation. In particular, this may be relevant for parents who are affected themselves by some of these adult diseases, but it is also accompanied by a large number of practical and ethical limitations. And there is now a continuous discussion between policymakers and professional societies about whether and how this technology should be regulated. Okay, so that's it. So thank you very much for listening.
Thank you, Shai. So Mara, can you open the sharing for me? So as we just heard from Shahar, how we can use machine learning for early diagnosis of Alzheimer, and then from Shai, how we can take computational approaches to predict whether a particular embryo has a signature that is associated with the trait of disease. These are wonderful examples for a potential power of computational medicine. I just wanted to use 2 minutes before we go to the questions to tell you about our main project in the faculty, and that is the new Center for Computational Medicine that we are starting these days in the faculty, which really aims to give us to provide a platform. The center aims to enable us to use big data, to use computational approaches in order to better fight diseases, discover how diseases work, diagnose them, and treat them. And it builds around the concept of bringing people together, doctors, researchers, computational people, theoreticians, giving them the data, giving them the tools, giving them the environment to really work together. So we're these days really establishing the new center. The center has these elements of acquiring data, analyzing it, using models for disease and research and applying it to patients, and also training people in medicine and computer science, like Dina presented in the beginning. And in the next few months, we're constructing a new building to house this. This is our campus in Ankara. This is an existing building, and this is going to be transformed to a 10-story building that will house all of this, house this new center. This is looking how it will, what it will look like up close, the entrance. And just to give you a sense, cutting through the building, it really employs all of these elements that we just discussed. We have teaching, we have innovation center, bringing discoveries to the clinic or to the public. We have a center for genomics, acquiring a lot of data, analyzing it, bioinformatics, and we have computational research floors and modeling floors to work on this. The center will be focusing on aging, on cancer, metabolic diseases, and infectious diseases. So all of the diseases that really trouble us today. And for us, for you being us, our community, we're looking for partners in the center. So if any of you want to get involved in thinking about the center and helping us establishing parts of it, this is where we really would like you to partner with us and be part of our community. So I'll stop here for questions. I just want to thank again Marco Canella, who's responsible for all of the Italian and the slides. It's super helpful, and I'm sure it helped you a lot. So thank you, Marco, and I think we should stop here for questions.
I understand that we have to start with the questions, and that means that the ball is to us, to Adriana and myself. Okay, good. First of all, thank you for this nice presentation. It was really very clear, and I think went already over some of the questions that I would have asked. That is to define better the use of artificial intelligence, terminology that personally I don't like because I think it scares people instead of providing support. But anyway, this is out of the discussion. One thing I would like to ask Dina is she mentioned the necessity to introduce in the computer a number of data, of any sort of data, the most complete set of data possible for the patient, to find the particularity of the patient and to define the treatment to use. Now, this would require an extremely organized clinic. How would you plan in the future to bring it into the territory? In other words, the physician who is following the patient usually does not have all this data. You have to test them. You need a big laboratory. So how can we spread this technology to the entire population instead of one specific center?
So I think that maybe the best way to answer this very important question is to give you an example, and I think that it will be clearer. Let's say we have a situation where a patient has a cyst in the pancreas. Very common phenomena. Many times it's found by routine MRI or routine CT scan, done for something else. Most of the patients, this cyst will stay there forever and nothing will happen. But small percentage of the patients will develop pancreatic cancer. So the clinical question that the physician in the clinic that you just described will have a patient with this cyst and will want to understand if his patient is in a high risk to develop the cancer right away and he needs to do a very big operation, or he can wait with it forever and nothing will happen. This is the clinical question. For that, you need to go through thousands and thousands of images of CT or MRI. You have to get all the information of many, many, many patients with this cyst. And then you put it all into the computer and let the computer think and come out with a algorithm or with something that is very, very using friendly. Now the patient come to your office, you put into the computer his details, and the computer will tell you what is the risk for this patient to develop cancer.
Yeah.
Okay, and then it's not that each and every physician will have to understand computational science.
That was not my, I'm sorry, that was not my question. I understand.
Sorry.
The development of the technology must be done in a very specialized center like the one you are building. But what I mean is, if you want to have my patient with that small cyst in the pancreas, you want to understand how it will develop, you need to test not only the imaging, but also the genetics of the patient, the composition of his microbiome, as was mentioned before, and so on. So these are data that the single physician, the individual physician does not have. How can you organize a network in which everybody can take advantage of your project? Will you have all the data put in a sort of cloud, iCloud, in which the physician puts in the name of the patient and then you decide what kind of test to do and centralize the diagnosis? This was actually what I'm thinking about organization, not the single patient, but the whole.
So I don't know the answer because I am not familiar in the details of computational science and how to save it. But I think that no doubt that in the era of AI and machine learning, we'll need such network that you're talking about.
Okay,
no doubt about it. But I want to tell you, Marco, the most important thing to understand is whether or not genetics play a part in the decision. We don't know it yet.
Yeah.
So you start by putting the genetic in, but maybe if you'll have a very accurate AI way to measure the cyst and the delta between two tests will be enough. Okay. We don't know it yet.
Okay, thank you very much. Thank you. Adriana.
Adriana, we cannot hear you. You're on mute still.
Okay, okay, sorry. I have a short comment and answer for Professor Ben-Yehuda. My comment is, okay, we had a vision of the future with this computational medicine, but my comment is, when at the moment we treat a patient, for example, for cancer, we treat this patient with the indication that we have from the traditional studies, from the traditional trials until the moment. So when I choose between, when I have to choose if I have to treat with a chemotherapy one patient or not treat with a chemotherapy, okay, I can have some tests until now that can give me a suggestion for this choose. But I think that the new medicine is the data medicine and computational medicine is totally different. I mean, and the professor told that we have to put a lot of things in a lot of big data in the computer, in the machine learning, and then we have to compare my patient with a number of indication from many patients to choose the right treatment. So I think that is changed the paradigm of the research. But until now, this is a future. But until now, we make decisions for treatment for the single patient based on the traditional research, right or not?
You're right. Unfortunately, our way to treat cancer in most cancers, not everyone, in breast cancer, it's very developed now. And you can send a sample of your patient to laboratory, central laboratory, worldwide, the foundation, and you get in the results of genes that are relevant for treatment, because
yes,
the fact that you find the herceptin, if you have a treatment that attack the herceptin, it makes sense. But if you find something that you don't have treatment for, it doesn't help at all. But in all other diseases right now, in most diseases, not all, but in most diseases, you are right, we are doing trial and error treatment. We give the patient the treatment, first line, he responds, fantastic. He doesn't respond, we give another treatment. He has severe side effects, we have to stop the treatment. So in many ways, we are not there yet, but that's the future and that's what we are aiming for.
So we have to change paradigm.
Right.
The research or
right.
the treatment. Yeah. Mm-hmm.
Right.
This is the future, not for now, at the moment.
In some diseases, you see, in acute leukemia, for example, each and every patient who comes into our department now, we do next generation sequencing, yeah, trying to look for mutations or for translocations that might give us a solution which treatment to use. Sometimes yes, we use for acute leukemia treatments that were designated to start with for other diseases, but we find that in the individual patient, it might help. It's again a trial and error, but it's more sophisticated. That's how we can call it.
Thank you.
Ittai, maybe you want to add some.
Well, I think just to, no, I think you answered well. I see a couple of questions from people in the audience. One is good for you, Dina, which is from Natasha Goro. Thanks for the nice presentation. How do you plan to deal with data privacy? Will you be able to share your very amazing data sets with data science around the world? Do you want to say something about this, Dina?
So I just want to tell you that it's an excellent question. I realized lately that the ethics in the AI era will be a whole new story. And we are now creating a new program for master degree in bioethics. It's not just the AI. You know that nowadays we can edit the DNA and the RNA. And when we are thinking about such procedures where we can correct DNA of patients or of fetus, we are thinking about very, very difficult ethical questions, because it depends in the hands of whom this very, very strong tool is now. So it's something that we need to start from scratch, understanding the ethics of the bioethics of a new era.
On the line of the question, I see another one that is also interesting in expanding the field. That says a question, how does the university tackle the ethical questions that arise from the new opportunities coming from these projects? Is it up to the university or at another level? And I would add, in case of some, you know that we are in an era in which there is a significant difficulty in rapport between physicians and patients, and quarrels, legal problems are arising every day. So who would be responsible in case of some mistake in the application of artificial intelligence? And again, going back to the question, is up to the university. As an academician, I would say that it is up to the university, at least at the first level. But
so the WHO created a new program to check these questions about ethics in the AI era.
Mm-hmm.
It will be something to do with the regulation and not in the universities, but the universities has to understand, the scientists, the physicians have to understand the real questions on the board.
And maybe give inputs. Maybe give inputs to the WHO.
Right, that's the idea. And in Israel, we also need, I'm sure that in Italy is the same, we also need to take into account the religion also, because some of the questions here has to do with religion. But for example, you know that when we are starting treatment to patients, we have to, the patient has to sign on informed consent that he agrees with the treatment. And I just found out that in the AI era, there is no way that the physician that will make the patient, ask the patient to sign on this informed consent really knows what he is signing on. The physicians.
Yeah.
And the physicians, maybe you can understand it, but also the computation scientists who develop the algorithm will not be able to understand how the machine came into the decision to give this treatment or not, because inbuilt in the program, there are what is called black boxes. So it's really complicated. And I think that the universities will have to study it and bring it to the regulation. And in our committee that prepared the master program, we have 2 rabbis that are professors of medicine. We have somebody from the Faculty of Law, somebody from philosophy, somebody from computational medicine. It's an interdiscipline committee that will decide what do we need to investigate regarding the ethics in the AI and the DNA, RNA editing right now. And I really think that it's a very important program, and I really hope that it will succeed, because it's something new that we are creating, and I hope it will succeed.
Okay, thank you. Adriana, have you any other question?
Yes, I changed my point of view, and I have another question. So Dr. Shahar mentioned a Clara study and mentioned about the collaboration between many research institutions over all the world. And I would like to ask if in these new programs for Embryos University, they have some programs with Italian university, because, for example, we do a research in this field. For example, in our IEO, in Institute of Oncology in Milan, or in University Bicocca in Milan, about this new field of medicine. My question is, what about in general, what about the collaboration with other institutions in the world in this particular moment, plus what if they have some collaborations with the Italian institution or universities in this field?
This for our research. So we have collaboration in Bologna.
Okay.
There is a big institute in Bologna, both in the University of Bologna, and there is an institute for neuropsychology and brain sciences. And the first studies that came, so we did a lot of studies there. 4 or 5 of them are already published. For example, one thing is that we did with the group in Bologna was to see how these orientations that they showed, how the strategy, how the brain deals with orientation in different age groups, like what happens in aging. In other is the relations, like how you can correct it with simple glasses. Like this is a little bit of a science fiction, but you can, since orientation is in the different domains, space, time, and society, or even politics. We speak about right and wing in politics. So maybe you can just put glasses on your eyes, that instead of showing you the center of the visual field, showing you the right, go more to the right or more to the left. So our collaborators, they're headed by Francesca Frasinetti from Bologna, but also Elisa Caramelli and Elisabetta and the students were also involved and visited us here, as did the professors. So what they did is like to take this and to do our test when you look more to the right or more to the left, and see how you can improve your performance just by this little small manipulation. So this we did in Italia, and for this in Italy, and for this we translated everything to Italian. I can add that one advantage, a huge advantage that we have in university with respect to companies, is that we are not making money. We want to advance medicine and we advance science, and that's why we are doing so many collaborations. And we have collaborations in the United States and in Germany and in France and in Brazil and in China, all over the world, because when people develop something, we can use it. And this is especially important for computational medicine, because in computational medicine, we create code. What that we create in the lab is code. We create a software. And if I have a code, so I can put it as a package of code and send it to somebody else, and then this somebody else just need to put the code in her code or in his code, and use it. So
it means that it is important to share the data.
Yeah. That's super important.
It's a very,
yes,
very, very important thing at this moment. You understand? Okay.
Yeah, and I see that
to collaborate.
Yeah. I see that there is a question about collaboration in France, and yes, about the Salpêtrière in the Institut. This Mr. Fitoussi knows of the place. ICM, the Institut de Cerveau la Moelle, Institute of Brain and Spine in Paris. So yeah, one of our collaborators there is Virginie van Wassenhof, and also in Salpêtrière. We are collaborating with Professor Daniel Margulis, and also with a group of Laurent Spinelli. So yeah, we have a lot of collaboration in Salpêtrière. The leading person in Alzheimer's in the world now that we follow is Bruno Dubois, is also in Salpêtrière.
I would like to ask another thing. I mean, this is about cooperation around the world on techniques. But you mentioned, not actually today, but in one of your leaflets, I saw that one that is a true advantage of the Hebrew University is that you're working on a rather selected population. But this means also that it will be difficult to expand to other realities. And I would say that Shai, Carmi mentioned before the difference of the performance of the test within your laboratory and when you applied it to other countries, and in Italy also you have a lower result, reliable result. How can we foresee for the future a cooperation that means having the same big data available and treat patients with the same specificity all over the world? I don't know if I'm
Is that a
clear.
question to me?
No, the question is in general for whoever wants to answer. I mean, I understand that really for research, it's beautiful to have a selected population, but in reality, population are mixed. And so if you want to expand to the clinical field and not only to research, you need to see how you can do it.
And I can say a few words from my angle, from genetic research. It is considered a big problem in genetics that we have that most of the data comes from the US or the UK, and there are continuous efforts to sorry. 1 second. No,
yeah, I will take it. I will take it for
sorry.
a second.
Yeah. There are continuous efforts to increase the sample sizes from different additional countries, including in Israel. There is a national project of the Ministry of Health to sequence the genomes of 100,000 Israelis. That is now at different stages. And the same, sorry, and the same for other countries. Okay. But that's someone else too.
I'm afraid that sometimes we are talking about two different kinds of data and data sets. One thing is to understand the disease with the old methodology of knowing the symptoms and so on. And one thing is to go further and to find if there is a genetical relationship, and through the study of big data, we can get to a more specific treatment for every patient. So two different levels of the discussion, I would say.
I can add 2 cents to the discussion. One is that it's difficult to do research all over the globe, but it's very to take blood and to do, but it's very easy to use to use software all over the world. And even if you go to the most neglected place on earth, you find people are going with their smartphones. And then that's what happened in our effort in Brazil, is that people we work with an NGO, with a non-governmental organization that said, well, we want to give health to people in the Amazonas. So we can't bring doctors to Amazonas, but we can bring tablets. So even from our lab's budget, we bought them some dozens of tablets, and then we trained them, and they took the tablets to the Amazonas. So this is something, and then you get the data in the Amazonas, and our algorithms send the instructions back to the people in the Amazonas according to what that is available here. So this is the first sense. The second is with a collaborator in London that we have. So we have data from 6 million people, 6.3 million people around the world. And then you can see what is common, because we are all humans, and what is different. And you can teach the algorithm to use things that are common because all brains are the same, and also to take under consideration, and the algorithm can do it. You can add as many parameters and many variables as you like. And so we also train the algorithm to treat differently people where this is also, you don't have to run so far. You can also speak about gender, because sometimes medicine for women and medicine for men is not the same. And also this has to be taken under consideration. We'll give just one example. In women, is much more important to treat diabetes to prevent Alzheimer's than in men. Why? I don't know, but this is proven.
Okay, thank you very much. This is very clear, and exactly that's what I meant as international cooperation. I see that there are a few questions. Again, two were left over from before, I think, and one deals with the initial discussion about pancreas. The question is if about this example about pancreas cancer, is it short-term or more? I think we can answer that this is just an example, and every kind of disease can be studied, and we still have to see if it is short or long term. But I will leave Dina to answer on this.
I think that I won't add anything to what you said. I just gave an example because it's something that we are studying in an interprofessional way. We have radiology, pathologists, genetics, and the basic science research. So it's just an example.
Okay. And the other question that has been unanswered so far is this kind of center already existing in the world, or you are the first one? I understand that there is something in New England. They think there is something at Cambridge, but I'm not sure if this is the answer they wanted.
First of all, I'm sure that there are centers like that building around the world, but I think that we bring something very unique, which is the all-togetherness.
Okay.
And I think that this is something that is very unique to us. Mm-hmm. And the other thing that I can tell you that we are pioneering is the computational medicine studies for students. So I think that some of the things that we are doing are very unique and pioneers, and some are less.
Thank you. I don't see any other question. I don't know if somebody else, I mean, there are different canals to receive the questions. Mara, do you have any other information, any question
no,
so far?
I don't see any other question, but whoever will think about another question, you are welcome to write me an email, and I will forward to Dina, to Ittai, to Shahar, or to Shai. So I think we can conclude now this webinar, and I would like to give the floor to Elisa Bianchi, our president of the Italian Friends, for a conclusion. And in the meanwhile, I thank you everybody for participating to the webinar.
Thank you all for the interesting discussion. Many compliments to the members of the Hebrew University for their amazing commitment. Tonight, both theoretical and practical aspects of computational medicine have been clarified. This is important to us all. I'm sure that the collaboration between Israel and Italy about this fundamental topic can go on fruitfully. And I hope and look forward to other exchanges of information in the future. Thank you. I wish you a good evening.
Thank you.
Bye.
Thank you.
Thank you very much.
Thank you. Goodbye.
Good night.
Thank you. Thank you, everybody. Bye.
Bye.